coronavirus could be defeated by 4 different methods. The first one involves developing a potent vaccine against the virus. The vaccine would induce the body immune system to fight the virus and produce antibodies that stay in the bloodstream for life. But developing vaccines under normal circumstances can take several years, as researchers must study the virus bit by bit in a detailed manner. Then they choose a small harmless fragment of the virus to serve as a vaccine. In other cases, they choose any fragment of the virus and deactivate it with chemical. The second method involves designing a drug that targets either the virus or the host cell. After the virus enters human cells, it begins making copies of itself. Then it destroys the cell and releases the new copies to infect new cells. The drug could block the virus from replicating inside human cells. But it means the virus would remain dormant without any activity inside human cells, during which it could mutate and come up with even more aggressive version of itself. The drug could also force the infected cell to kill itself by apoptosis. Apoptosis is a process in which the cell releases molecules that eat it from immune cell called phagocytes, then come it ingests the cellular components. Developing such a drug normally takes 14 years of research, development, and testing, which makes this an unlikely solution on the going global outbreak. The third method involves testing existing drugs to see which one could work against the virus. Viruses in general share similarity in their genome can be high as 90%, which means existing drugs for other viruses such as influenza virus or HIV could solve the problem and help contain spread of coronavirus. Such drugs could target the same types and block the same processes in viral replication. Current reports show that anti-HIV drugs are being repurposed to fight coronavirus. The two drugs have been around for 10 to 15 years and are off patent. The fourth method involves genetically engineering human T-cells using CRISPR, and then injecting the genetically engineered cell into human body. The T-cells are engineered to become more potent, are recognizing and killing the infected cells. Genetically engineering human T-cells must be done separately and differently for each person because our genomes are slightly different. The engineered T-cell won't work on everyone, which means everyone infected must be his or her own genetically engineered T-cells. Given how fast the current outbreak is spreading and how slow genetic engineering, this was likely an ideal solution for our ongoing outbreak. For the moment, our only hope is that a vaccine is developed fast enough to contain the disease, or an existing drug be repurposed to block the virus from spreading further.